Now, these stem cells can be used to replace damaged cells. It can repair tissues, and we use this in tissue engineering. Reg, so as jy kyk, hierdie stamcelle kan gebruik word vir terapeutiese kloning. Nou wat is terapeutiese kloning? Terapeutiese kloning is waar ons byvoorbeeld vir terapeutiese doeleindes om iets te herstel, to fix something, to repair something. Now stem cells, this history of stem cell and the research on this has started very long ago. As you can see, in 1938. I'm not going to read through this, but just to show you that this has come along for quite a long time. Eers in 1997, by die Rosalind Instituut, was die eerste skaap met die naam Dolly gekloon. En nie uit 'n eiersel uit nie, maar uit 'n gewone borsel van 'n ander skaap. And this is somatic cell nuclear transfer or cloning as we know it. Now, how is this done? Once again, it's steps. So I'm just going to go through this for you. If we look at this, I'm going to use Dolly. They have taken a breast cell of a 6-year-old sheep, a borsel van 'n 6 jaar oude skaap.
Die eier, né?
Ja, uit die eier uit.
Daar's plaaskinders daar buite.
Ja.
Eier van die skaap, ja.
Reg. Wat het hulle met hierdie borsel gemaak? Hulle het die kern daaruit verwyder. Sien jy, die kern het hulle verwyder. Hulle het 'n sigoot geneem, 'n eiersel geneem, en daardie kern, jy sal sien dit is 'n leë eiersel. Met ander woorde, hierdie ovum, they have taken the nucleus from this and they have placed that nucleus from the breast cell into the ovum, and this is what they had, and they gave it an electrical shock, and that's a very important thing because it had consequences. So by doing this, they had 434, not that you have to know this, successfully reconstructed eggs, but just to show you how many times they have tried. From this, they only had 29 reconstructed eggs, en dit het hulle geplaas in halfswanger skaap en pseudo-pregnant sheep. Now, what is a pseudo-pregnant sheep? Lorraine. If you can think of this, those of you who have done fertilization and reproduction in humans, you know that no egg can be accepted in the uterus if the uterus lining or the uterine lining, the endometrium, is not prepared for this. So a pseudo-pregnant sheep is where they injected hormones into those sheep to prepare them to accept this egg, this fertilized egg. And this, in this instance, it's not a fertilized egg, but it's the nucleus of another cell that was placed into the egg. And the only successful cloned sheep from this was Dolly. So from 434 of these cells, you see, and this process was done to start mitosis.
And this is quite interesting.
Very interesting.
Yeah, is that they do have to give that little bit of an electric shock. And I think some of the learners might have seen old Frankenstein movies.
Yeah.
Yeah, where they put the parts together from, they take from bodies from coffins and then also send an electric shock through it, and then you get Frankenstein. Of course, it's not remotely anything like that. Don't think it. I've actually had one of my learners ask me, so that's, I don't know if other people have the same idea. But it's very important, that little electric shock, because as Karen said, that starts mitosis. In other words, it starts the embryo to grow. Why do they put the nucleus of the mammary gland, so the mammary cell, into an ovum from which the nucleus was removed? What can be the use of that, or the reason for that? Why not in any other cell?
I think in science, in science, it's very important that we understand why is it that we do things. And this is also that you have to concentrate on things like this, because these are the types of questions that you can expect. So just think about it. They have taken a normal nucleus from a normal cell and placed it into an ovum.
Onthou daardie ovum se inhoud is ook nog die organelle wat ook hulle eie DNA het ook nog nie oud geword nie, is nie beskadig nie, daar's nie chemiese stowwe nie. So the ovum is actually also the best that could be given to that new nucleus.
Ja. En as ons nou praat van hierdie eiersel of die nou nie die eiersel nie, maar die eier, die borsel, dan moet jy weet dis 'n gewone liggaamsel. Dis 'n somatiese sel. And what does this mean? And if you can look at the slide there, it means that the DNA of a somatic cell still contains all the information needed to direct or to form an entire new organism. A normal somatic cell was used to form a new or an entire organism. And I think that is the importance that Lorraine wanted to point out to you. So onthou nou dit. 'n Gewone somatiese sel was gebruik en dit het oorsprong gegee aan Dolly.
En wat belangrik is dat hierdie somatiese sel is met ander woorde dit is 'n sel wat uit 'n eier uitgevat het. Daardie sel het nie so geword dat sy DNA nou net eierwerk kon doen nie. In other words, we can also take you can take a cell from our upper arm, which means that if that cell was used in a cloning, it would not just form upper arm skin DNA or directions for that cell to do it. So you can really switch on the DNA again, so that maybe that nucleus can direct the cell to become other organs, other tissues. And that's really an important aspect.